Thank you for attending the session. I'd like to present you the activities of Regenxbio. We're a gene therapy company using AAV vectors. This is our forward-looking statement. We're a public company. We like to see ourselves as leaders in AAV gene therapy. We have 4 clinical stage programs today, and all this is based on proprietary technology that we have. We call this the NAV technology platform. It's a collection of AAV vectors for which we have exclusive rights, and that includes AAV 7, 8, 9, 10, RH10, sorry, and a number of others as I'll discuss. We have a number of licensees that are also using this technology that has led this year into a landmark first FDA-approved AAV9 product for SMA, Zolgensma. And there are about 15 clinical stage product candidates that are being developed by third-party licensees here. That amounts to a total of about 20 partnered programs in total. So just a few words on our technology platform. This is based on the work that happened in the early 2000s at UPenn. Guangping Gao in the laboratory of Jim Wilson discovered basically a trove of new naturally occurring AAV sequences in primate and non-human primate and human biopsies. This represents the family tree of all these capsids. And RegenX now owns exclusive rights to all these capsids, and there's a portfolio of about 100 patents and patent applications worldwide. This IP covers the vectors that can be packaged into these different capsids. So there are different families of these capsids with obviously different properties. It also covers capsid sequences which are close to these capsids within 95% of identity. So this collection we have serves to us as the basis of developing novel capsids using this biodiversity. And we were actively working at that as well. And that starts actually by characterizing all these families. Only a handful of them have been today fully characterized and are being used in the clinic. Importantly, we have developed a end-to-end commercial scale capability for GMP production with a number of these vectors. And obviously for those that we use in the clinic, those are our lead programs. Today I'll get in more details into our retinal disease program that we call RGX-314. This is for age-related macular degeneration, the wet form, but we're also applying the same vector to a number of other retinal diseases. Beside these main programs in ophthalmology, we have programs in the clinic in neurodegenerative disease, targeting lysosomal storage disease, MPS 1 and 2, Hunter-Hurler disease, as well as coming up for an IND submission, Batten disease, CLN2. Recently, we've started a research collaboration with a Swiss company called Neurimmune, who's an antibody designer and maker, vectorizing. The goal is to vectorize using AAV those antibodies, and the prime targets are here tauopathies. We also have a liver-directed activities in the clinic. We have a trial going on with using AAV8 in homozygous familial hypercholesterolemia. And we also announced a research program that we initiated recently on hereditary angioedema. But I'll focus for the rest of my talk on our main program, RGX-314, for the treatment of wet age-related macular degeneration. So the disease, to be very quick, is blurring of central vision, and that's due to neovascularization that occurs, a formation of leaky blood vessels that basically ruins the architecture of the retina around the macula and prevents vision. There is, I mean, for a number of years now, there have been treatment for this disease that used to be totally intractable, and anti-VEGF proteins have reached the market, and they are definitely a great help for this patient that eventually recover vision. But the problem with the treatment and the unmet medical need that we are addressing with gene therapy is that especially in this population of aged patients, compliance is a big problem. And unfortunately, if you miss one of your regular injections, often monthly injections in these patients, the vision will go down, and there's no way it can be it can be recovered. So although the standard of care are pretty efficient in a well-controlled population that's fully compliant, the compliance problem really create an issue for this population. So it's a large population, over 2 million patients estimated in the US, Europe, and Japan. What we're doing is we're using AAV8 because it's been demonstrated over and over again that AAV8 is actually a very powerful vector to reach retinal cells, which is where we want to express our anti-VEGF molecule. And by expressing this molecule constantly, in a one-off administration, we allow for the production of the therapeutic protein. So we're using a subretinal route of administration for injecting our AAV8, and this is because this is the most efficient way of reaching the retina. Obviously, we're just injecting where the cells are, and so that's why we've chosen the subretinal route. We're also working, and I'll say a few words, on another route of administration to reach the same space in the retina, but coming from the other side, coming through the suprachoroidal space, and I'll say a few words about that. So this trial that I'll describe to you is based on a lot of preclinical studies. We've built a vector that expresses a fragment antibody against VEGF, and this is an antibody which is pretty much analogous to ranibizumab, also known as Lucentis. And in a single AAV construct, we express both heavy and light chain, and they associate, and they make a product that's active and that we've thoroughly characterized, including in primate studies, which is the lower part of this slide here, where we show that we can obtain a dose response on the left panel at 4 weeks in monkeys, and that constant production of the protein can be measured very long term, and here in this study, over 300 days. So the trial structure is explained here. It's a dose escalation trial, and we've now dosed 5 cohorts. We've completed the trial. Altogether, we've dosed 42 subjects. All these subjects are first tested for their response to anti-VEGF. That is, they come in, they're injected with the standard of care, and their response is measured after a month, and those who are responsive are included in the trial. So after having received AAV, they're regularly followed up every month by their physician, and the physician can actually decide to re-inject with the recombinant factor if something goes wrong, if fluid comes back in the eye, or if there is something that he considers is a problem. After a year, we look at, after 26 weeks, we've looked at safety data, and secondary endpoints are being measured at 54 and 106 weeks, and those secondary endpoints are vision and thickness of the retina as measured by OCT. So the safety and data summary is that basically in all these patients, 42 patients, the treatment was well tolerated at all doses. We haven't seen any drug-related adverse events, severe adverse events. Importantly, we're not observing, using this subretinal route of administration, any immune response against the treatment, any inflammation, and I have to say that those patients are not screened for pre-existing antibodies against AAV8, right? So we choose a route of administration that really isolates the treatment from immune responses. We see, importantly, a dose-dependent increase in the protein expression level that we can measure using an aqueous chamber tap in this patient. So we can document that our transgene is active and makes the protein, and this is dependent on the dose. In dose 3, as I'll show you, we start seeing signs of efficacy with half of the subjects now being free of injection after more than 18 months. Free of injection of the standard of care, that is, they don't need to get their monthly treatment anymore. That's a summary of the data. The two first columns of these tables are protein expression at 1 month and a year, and you see that there is this dose dependence. Then we look at the number of injections per patient. If you look at cohort 1, patients have an average of around 10 injections per year, which is roughly once a month. Basically, cohort 1, we don't see much change from the regular population. Slight increase with cohort 2, but with cohort 3, we have a drastic reduction in the average number of injections per patient. And that goes with improvement in retinal thickness as well as vision measured in BCVA. So now this is cohort 3, only the subjects which are doing good, that is that they've never been re-injected with the standard of care over the past 18 months. This is the data at 1 year. You see that on the average, they recover 10 letters of vision, and their retinal thickness goes down by 59 microns on average. And this is another representation. Those are the 3 doses for BCVA, vision, and retinal thickness. I'm not going to go into any kind of details, but if you look at the data on the right, again, we have a good improvement in vision for cohort 3 and a reduction in retinal thickness. So what I just showed you, I mean, encourages us to move forward. We're going to release the data for the remaining cohort 4 and cohort 5, which are higher doses, and that will happen next week at the conference of the American Association of Ophthalmology. Unfortunately, it's bad timing because I can't tell you anything about this data, but we're doing it next week, so if you're interested, look it up. We also initiate now a phase 2B trial with RGX-314. And we're also looking into using the same vector for another application, which is diabetic retinopathy. And we want to initiate a phase 2 study in DR by the end of this year. The last thing I want to mention is our investigation of suprachoroidal space delivery for AAV. The reason why we're doing that is that eventually this is a way to administer without having to get into the surgical suite, right? This is eventually an in-office procedure, and our initial data that we obtained in collaboration with Peter Campochiaro at Johns Hopkins shows that in non-human primates and a number of other animals, using this space with AAV8 it can be very efficient at getting at the right cells in the retina, that is the RPE and the photoreceptors. And we've used our RGX-314 vector to show that we can get good concentration of the protein in this animal model. So that really encourages us to look deeper into that and see how much we can actually use this route of administration in the future for delivery of our vectors. And on that, I'll thank you very much.